co-host on YouTube. Supposedly is how it works.
We are live.
Okay, we're live on YouTube.
Yay. Co-host on YouTube. Supposedly is how it works.
We are live.
Okay, we're live on YouTube. If anyone who's watching on YouTube who's registered early, make sure you use the other link, not just watch on YouTube. I wish there were a mute button for my office phone. You don't know how many times my phone has rung while I'm in the middle of recording a lecture or having students on Zoom.
If we had like an actual modern phone instead
yeah, exactly. I mean, at least it doesn't have a dial, right? It actually has buttons.
It's like one step above a rotary phone, really.
So was a live on YouTube a Zoom function? Good to know.
Okay, that's better. I was getting a lot of weird feedback. I was like, oh, I'm hearing echoes. So I just muted the YouTube so that I can only hear what's going on here. So we're good now, I think. Ooh. So in 5 minutes, so we have 6 people who are already watching on the YouTube.
All right.
So those of you who are on YouTube, go ahead and like introduce yourself if you want in the comments. We'll be there watching. I'm totally logging out of my email because, of course, we hear the dings.
Oh gosh, good point.
Mm-hmm. All right, anyone on YouTube, if you can tell me you can hear us fine, it'd be really great. If you can tell us in the comments, that'd be awesome. Or in the live chat. You registered for the event, but can't find the information on the Zoom. Is there a separate email for this? If you registered online and you got a confirmation email, then that means you successfully registered and you were within the 300 limit that we did have for the meeting. If you did not get a confirmation email, then you have to watch the YouTube video instead. Awesome. Thank you for telling me you can hear me. Yay.
And when did you last send that link for a Zoom?
You get a confirmation email with the Zoom link. And it's an independently designed link for each person who registered. So it's not a specific, it's not a specific.
Okay, Zoom. Fair enough.
Okay, looks like we got 3 minutes till.
Is our guest speaker here yet?
Well, I am searching. I'm still waiting to see if he's in. Okay. All right, let me check something really quickly. That is interesting. I'm reading. There he is. Hey.
Hey there, thank you so much for helping us out with this. I really appreciate it.
Yeah, I'm really excited. It's going to be fun. I don't know if can you hear us?
Okay, cool. We're just
All right.
checking.
I think I'm going to start letting people in. It's
Go
3.
ahead.
What are you guys thinking? Start it 3 or 5 minutes after? You've got a lot of people to let in. Ashley, it's your call.
They're already being let in, actually.
Okay. Well, I can't see what you guys are looking at, so tell me when you want me to start. Oh, I guess I can see.
Wow. Oh my goodness. I don't even know what this looks like on the multi-grid thing, but wow. So, wow. Thank you all for being here. This is awesome. Okay, we're gonna get started in about a minute. There's some people who are still joining us, and we also have some people in the live stream. I'm sorry if you can hear yowling. That's actually my cat. He's in the bathroom. So
Oh no, he locked him away.
he was bad. He was messing with the Christmas tree. So exactly. See, yeah, Eve knows. Okay, and I'm going to. All right, well, we'll go ahead and have Dr. Wendy Hood introduce DEI. We're going to go ahead and get started. So, Dr. Hood, if you want to introduce what DEI is.
I will. So it's my pleasure to welcome you all to the inaugural OurSTEMstory presentation and discussion. My name is Wendy Hood. I'm an associate professor here in biological sciences at Auburn and the chair of the Diversity, Equity, and Inclusion Committee. So the unfortunate and untimely deaths of George Floyd, Ahmaud Arbery, Breonna Taylor, and so many others, and the Black Lives Matter movement have both been a wake-up call and highly motivating for many faculty and students in our department. And we realized that we could be doing a lot more to ensure that our department is a welcoming and inclusive environment, not only for people of color, but anyone belonging to an underrepresented group. So at the beginning of this semester, we formally formed our DEI committee. I'm really pleased. Our committee includes 23 individuals, including faculty, staff, graduate students, and even an undergrad. The committee is divided into 8 subcommittees, each taking on unique challenges to improve diversity and inclusion in our department. So those subcommittees include an undergraduate recruitment committee, a graduate student recruitment committee, activities organized by the Biological Sciences Graduate Student Association. We have a group that focuses on educating our students in the classroom about the contributions of underrepresented groups, and they discuss or tackling inclusion type topics. We have a workshops committee, a committee that is building our webpage and working on our social media presence so that we can make our activities more forward facing. We have a group that's charged with bringing people in our department together, particularly those from underrepresented groups, so that everyone feels part of our biological sciences family. And finally, one of our subcommittees is charged with organizing public discussion that will both critically evaluate the ethical missteps of scientists and physicians that have taken missteps they've taken in the past, an effort to acknowledge the past and avoid similar mistakes in the future, as well as recognize the contribution of underrepresented scientists in STEM, which is science, technology, engineering, and math. So it was this charge that gave rise to our STEM story. So based on the number of people who are joining us today, so we have almost 220 people who are online. It's clear to me and the rest of the committee that these are much needed discussions. So thank you all who have joined us today. And I'd like to thank Ashley Williams, Corey Black, Rita Grays, and Marie Strader for their hard work organizing the OurSTEMstory program. And I particularly like to thank Dr. Ram Seshardi for joining us today.
Right, thank you. So I'm just going to do a quick little overview of our program. And so some of you might have gotten the email a couple of days ago, if not on Sunday, of the actual program we have set for you all. And I'm uploading it right now to the chat. It's a little easier to do this on Zoom because I can just share documents with you instantaneously. And so if you look at the event program, obviously we're going to start it with Dr. Wendy Hood giving a greetings and an overview of the DEI, or Diversity, Equity, Inclusion Committee. And then I'm going to talk to you for about 2 minutes about the program structure and some of the etiquette that we are expecting from everyone who's participating. And directly after that, we will have a pre-assessment, a concept check pre-assessment, where we'll ask you a few questions based on what you already know regarding intellectual property and the Watson and Crick versus Rouse and Franklin case. And we'll have Dr. Rita Grays, who's going to introduce the topic of intellectual property, which is the research ethics principle of interest for this entire meeting. And then, of course, we'll have Dr. Ram Seshardi, who's going to explain a little bit more about the experiments that were done in order to understand the structure of DNA and the contributions of Dr. Rosalind Franklin to our understanding of the structure of DNA. And then we'll also have a Q&A and discussion where we'll allow you guys to ask questions with the speaker, or to just simply contribute some level of some of your knowledge to the topic of interest. And then we'll have a post-assessment exam, or post-assessment questions. You'll simply answer the same questions, and we'll just do a comparison to see if you guys change your responses after the information is presented to you. And then we'll have some closing remarks and a dismissal. The entire meeting is expected to last for 1 hour and 30 minutes. So to begin with, some etiquette rules that we do have for you guys. Please be respectful. That means no explicit content is allowed. This is live streamed. And if you're uncomfortable with that being live streamed, you can turn off, if you're uncomfortable with your image being placed on a public platform, go ahead and turn off your videos now. But you can leave we'll ask you guys to unmute yourselves if you have specific questions during the discussion period. Do not unmute yourselves unless given the authorization to do so. We do have it set so you cannot unmute yourselves without our permission. During the discussion portion, you should be able to raise your hand with an icon. So if you click on the little icon and you raise your hand, then we'll allow you guys to ask a question to the speaker during the discussion portion. And If you are live viewing at the moment and you would like to ask questions on your own, you want the speaker to answer your questions, go ahead and ask your question on Twitter. You will use the hashtag #OurSTEMstory, and then we'll allow those questions to go to our moderator, who will then ask the question to the speaker on your behalf. So you can also participate. For the chat, if you do have questions, but you're not comfortable with asking those using the video, then you can go ahead and put your questions into the chat. Those will go directly to the co-host for the event, and then that'll go to the moderator, who will then ask the question on your behalf. Okay, so we are going to get started first with our pre-assessment. There are 5 questions on IP, or intellectual property. The last question is kind of a opinion question, and it's the general question that's being posed for the entire meeting, is whether or not Watson and Crick did commit intellectual property theft against Dr. Rosalind Franklin. All right, so right now I'm gonna give you guys a few minutes to answer these questions on our pre-assessment polls, and you should be able to see them right now. And if you can't, just go ahead and let us know in the chat, and we'll try and figure out if we're gonna make this work for everyone. Okay, we'll just give you another 30 seconds. We'll give you until 2 minutes, and then we'll go ahead and have Dr. Rita Graves go ahead and do her presentation introducing the topic of intellectual property. Okay, Rita, if you'd like to go ahead and get started.
All right, thank you. Hi, everybody. Give me a second to go ahead and get my slides up. Okay, all right. Can you guys see my slides? You can hear me? All right. Okay, so I'm Rita Graves. I am an associate professor in the Department of Biological Sciences here at Auburn. And I would like to join the other organizers in welcoming you guys to OurSTEMstory. Thank you. So today, Dr. Seshardi is going to guide us through the story of Dr. Rosalind Franklin's work and her role in the discovery of the structure of DNA. And we want to ask the question, did Dr. James Watson and Dr. Francis Crick commit intellectual property theft when they published their seminal work on the structure of DNA? And so each OurSTEMstory session is going to start with a lecture introducing the ethical concepts and terms, and that is meant to provide a foundation for the speaker's presentation. So that's my role. And for context, I usually teach introductory genetics, and I teach a lab on genetic engineering. So I'm very familiar with the structure of DNA. Teaching the ethics of intellectual property is something that's new to me. So I'm learning new things, and we're all learning together. So again, our goal is to provide context for that central question regarding intellectual property theft in this case. And so an easy place to start is to define intellectual property. So what is intellectual property? And so all of us could probably write down, our personal definition of IP. If you're not familiar with it, you could also just look at the words and kind of realize, okay, intellectual, it's something to do with thinking, with creativity. Property, it's about ownership. And you'd come pretty close to a good definition of intellectual property. But for the OurSTEMstory series, what we want to do is build on a foundation of terms, definitions that have been put together by the National Institutes of Health. And so you can find this in their bioethics glossary. And there they define intellectual property as legally recognized property pertaining to intellectual activity, such as creative works or inventions. And so we have not only this idea that this is property, something you own, that is a product of intellectual labor, intellectual work, but also that it's legally recognized. And so when we think about intellectual property and intellectual property rights, these are very tied into the protection of those rights under the law. So very briefly, we can think about what intellectual property law actually protects. And so we start from the ideas, and those ideas need to be expressed as something tangible. So we're protecting tangible, realized expression of someone's original ideas, and we're protecting ownership by the inventors and creators of those ideas, of control over the expression of those ideas. And so at a very fundamental level, this is a legal expression of an ethical concept, which is that if you thought of the idea and then you did the work that was necessary to realize it, to make it tangible, then you're the one who should have ownership. You should get to decide how that idea is used, how it's expressed, and you should have commercialization rights. But if you start to dig into IP law, what you see is that something that's very simple is actually very complicated in terms of the body of law that controls protection of intellectual property rights. And so each kind of intellectual property actually has a different mechanism for protecting that IP. So creative works like novels or paintings are protected by copyrights. Business information is protected as trade secrets. Slogans and logos are protected by trademarks, and maybe most familiar to scientists and relevant for scientific research, inventions, discoveries are protected by patent law. And in each of these cases, the law has to take something, some intellectual property, and then decide whether that is something that can be owned, can be protected under this law. And so that kind of boils down to four sort of fundamental questions about the application of intellectual property rights and the definition of intellectual property in law. And so these are things like, is the idea truly novel? Is this the first instance of the idea? And so I don't know about you, but I come up with $1 million idea literally all the time, and I'm going to be a very wealthy woman right up until the moment where I Google the idea and I see that, somebody else has gotten there before me. So you need to establish that it's new. And the other thing is that a lot of the things that we think of, like inventions and machines and new drugs, it's not just one idea. They're really a whole set of different ideas that are put together and expressed as one thing. And so the law needs to decide, is the ownership over all of that kind of single expression of an idea, or is the ownership that's being established over a very specific piece that's integral to that larger expression of the idea? You also need to ask, is the idea actionable? So this is another one that's preventing me from being a millionaire. So I can give you all for free a really amazing, excellent idea, which is that we should genetically engineer horses and make unicorns. So I think that if you could do that, you could make a lot of money. But at least right now, that idea is not actionable. And so my idea would not be protected under IP law. And finally, I think it's important to realize that there may be conflicts between intellectual property rights and other kinds of rights or other interests of society at large. So there may be certain ideas that as a society, we decide that no one should own for the greater benefit of everyone makes up that society. Or you might think that you might view it as that is something that is owned by everyone instead of one particular person. The other thing is that once you get beyond these basic questions, conflicts over intellectual property can be quite complicated. And there are many other questions that we might want to ask. And here we want to center ethics. So we want to be thinking about respect for persons, principles of fairness, maximizing benefit while reducing harm. As we think of, what are the, who should get credit for an idea if there's a conflict over who should have the intellectual property rights, intellectual ownership over that IP. What form should the credit take? What's the relevant evidence? How can we establish a chain from the person who originated that idea and first expressed it as something tangible and link those two in order to assign credit? We can ask things like who or what affects the way the conflict gets resolved, and that might be in the court of public opinion or in a court of law. And there are things like how is the case of each side presented in court? How are the individuals that are involved in whatever this conflict is telling their story in the media? All of those things might affect how a conflict gets resolved. So we have our definition. We have some basic questions we can ask about IP when determining whether it's something like who owns it. And we have a set of ethical kinds of questions we can ask when we're thinking about respect for persons and principles of fairness. And so the first thing I want to do is just give you guys an example of some intellectual property. And so this example, the creative work is actually a dance, and it's a dance called The Renegade. And this became just a huge viral sensation on TikTok. There were all of these influencers and very well-known celebrities, not going to name names, but some on this slide, who did this dance and popularized it. And as we kind of all know, in this day and age, it's not just for fun. Internet content makes money. And when they did this, there wasn't any credit established. It was just sort of treated as coming from the kind of amorphous ether of the internet. And so this was like all over the place. But in fact, it has a creator. So somebody created this, designed this dance. And early this year, a reporter from the New York Times actually looked into this and found that this dance that had just gone wild on the internet had been created by a 14-year-old girl named Jalaiah Harmon. And she had posted it to a different social media platform called Funimate. And it was then lifted from that platform onto TikTok, where it was popularized by more well-known influencers and celebrities. And so this is a fun case to look at, number one, because it has a happy ending. So her credit was reestablished by the New York Times article and eventually on the Ellen show. And you should Google that because it's really wonderful to watch her appearance on Ellen. And she's done all kinds of other appearances. And she's in fact working with gaming companies like Epic Games to translate the dance into an emote in the video game. So all of that is great. But we can think about these basic questions like, is it a novel idea? Had anybody come up with this dance before? What is the actual IP? Like what form does it take? So for a dance, that's actually choreography. And, who should get credit? And is, we want Jalaiah to be able to control, how this idea of this dance, the choreography is used and have commercialization rights and all of that. And in this case, we see it's been resolved in her favor, which is great. All right, so this is something from popular culture, but we want to bring this back and talk about IP rights and IP law as they apply to scientific research, to science. So when it comes to science, we can start by asking who owns science IP, meaning intellectual property that's produced in the course of scientific research. And you might think back to my earlier slides and say, aha, I know the answer to this question because I know who does scientific research. That is done by scientists, and therefore scientists should own science IP. But in fact, ownership of IP that is produced in the course of science is very rarely owned by scientists. It is mostly owned by other institutions like companies and universities. And so this is kind of easy to think about when we think about companies. So a company like Pfizer employs scientists in their R&D department, and they produce something like a vaccine. And so that IP is owned by Pfizer. It's not owned by the individual scientists. And it turns out that universities kind of work the same way. So when you're employed by a university as a scientist, most of the time you'll sign a contract, which is just essentially says that anything you do while at the university using university resources is in fact, that IP is owned by the university. And that actually started as a practice very early on in the 1920s, and it is basically the norm. So when we think about who owns science IP, most of the time companies and universities, very rarely the scientists themselves. Now there are contracts, between the universities and the scientists, so they get some of the credit and they get some of the benefit of commercialization. They're just not the owners. We can also ask, what are the kinds of products of scientific research that IP law can assign ownership of? And so there's a lot of things, kind of typical things like, the writing that makes up scientific publications, figures, concepts, formulas, designs. But there are some things that are produced in the course of modern scientific research that we're kind of not used to thinking of as being owned in this way. And so, for example, today scientists might genetically engineer a bacterial strain to do a specific job, like help break down plastics for recycling of plastics. And IP law can assign ownership over that bacterial strain, that particular genotype of the bacteria. And so IP law not only can assign ownership over images and writing, but also of living things. And that kind of brings IP law and IP rights into conflict with other human rights because, human beings are living organisms and their tissues and cell lines are living things that can be changed in the course of scientific research, and in fact, they can be owned. So there's a established body of law which has looked at this conflict and basically resolved it in favor of the IP rights rather than the rights of an individual to control what happens to their tissues and to cell lines that are derived from those tissues. And if you read these decisions, it can be quite disturbing. So there's an example here, which is part of a very influential case. And this is the majority opinion from the California Supreme Court in the case of Moore versus the Regents of the University of California. And this is one of those cases where there was a conflict over the intellectual property rights to a cell line that was derived from Mr. Moore's tissues. And so as part of that decision, they wrote, an individual has no proprietary interest over removed body parts. So you don't have any interest or control over what happens with your own tissues or cell lines that are derived from them. So this is actually like a really crucial part of another story that went untold for a long time, but has recently become better known, which is the story of the HeLa cell line, which is just, used in all kinds of medical research and in science and scientific research in general, and is derived from the tissue of Henrietta Lacks. And that was done without her consent. And so we are actually going to tell that story as part of OurSTEMstory in fall of 2021. And we're going to have a family member come and speak and guide us in that story. So if you're interested in that, set a reminder, and when it comes around, register and be part of that discussion. Okay, so we've talked about the definition of IP. We've talked a little bit about how IP, who owns science IP and what kinds of things can be owned. And so I want to give an example, a science example of some intellectual property and of a conflict over that intellectual property. And that intellectual property, that idea, is CRISPR-Cas9. So CRISPR-Cas9 is a technology, a molecular tool set that allows scientists to make very specific changes in DNA sequence. And that means that you can change gene sequences, you can change how genes are expressed, and you can change the associated traits, like eye color or some kind of genetic disease like sickle cell anemia. And so that is, a very profitable technology. And as you can imagine, when something is extremely important and profitable, you can get a lot of conflict coming out of that. And so you have different kinds of, we have our idea, there's different ways that credit can be assigned, and there's this underlying conflict. So very recently, this story has become easier to tell because we have a very clear assignment of credit in perhaps kind of the most prominent way that the scientific community can acknowledge someone's credit as the discoverer of an idea. And that is that the Nobel Prize in Chemistry in 2020 was awarded to two scientists, Dr. Charpentier and Dr. Doudna, for the development of a method for genome editing, which is CRISPR-Cas9. So Dr. Charpentier and Dr. Doudna took this system, CRISPR-Cas9, that's a natural system that exists in bacteria for defense against viruses, and streamlined it and adapted it as a molecular tool set for gene editing, which they then applied in bacterial cells. And so here they're credited as the originators of this idea, this technology, CRISPR-Cas9. But when we think of other ways that credit can be assigned, such as commercialization rights, commercialization rights are not assigned to these scientists. And this is still in the courts, okay? But many of the decisions have actually come down in favor not of Dr. Charpentier, it's hard for me to say, I'm sorry, or Dr. Doudna, and not the institutions that they're associated with, but in fact, a different institution, the Broad Institute. So how did this conflict arise? Well, part of it is that there's lots of scientists that have worked on this question. There's scientists who worked on CRISPR-Cas9 before Dr. Charpentier and Dr. Doudna. And there's scientists that worked on it after them or in parallel with them, taking that original technology and applying it to different contexts. And so there's another group who published shortly after the original publication from Dr. Doudna and Dr. Charpentier, who, Dr. Zheng and Dr. Church, they took CRISPR-Cas9 technology and they applied it in eukaryotic cells. So why is that important? It's important because human beings are eukaryotes. And so doing genetic engineering in our cells is that application, using CRISPR-Cas9 to edit DNA in eukaryotes. And in addition to these scientists, we also have some other players in this story. And this maybe has to do with why we have all of this kind of conflict over the IP rights, because each of these scientists works at a particular institution or university, like the Max Planck Institute or the Broad Institute or UC Berkeley. And in fact, each of these scientists, before these original papers were even published, was involved in the founding of different companies, each of which had the mission to apply CRISPR-Cas9 technology to things like therapeutics, medicine, and agriculture. So very profitable applications. And as we talked about before, in general, it's not the scientists that own the IP rights, but it's the institutions. So now we have a conflict, and we can kind of think of the kinds of questions that we might ask when we think about how it should be resolved. And so one is, what is the relevant evidence? As scientists, we're kind of lucky because the relevant evidence is actually very clear, and it's not in dispute by any of the parties because we can look directly to the scientific record. We know who published what pieces of this technology and when, and that is very clear. So we know the role that Dr. Charpentier and Dr. Doudna played, and we know what Dr. Zhang and Dr. Church published, and we know in what order those papers came out. But there's still a conflict, and there's still kind of differences in the way that the different players and institutes that are involved tell the story and view the story. So now we have maybe not the evidence, but the kind of way that that evidence is presented in media by the different people. So the scientists are actually, they're not very aggressive. They're pretty straightforward in what they say about it when they're asked. Each of them has a slightly different style. So Dr. Doudna has been, fairly modest, saying, look, we thought it would work in eukaryotes, but we weren't sure, and we hadn't actually applied it in that system. Dr. Charpentier has been very direct. Like, you're going to ask me about CRISPR, I'm going to say we did it in my lab with Dr. Doudna. That's the end of the story. Dr. Zhang, again, coming back to the specifics of not just what is the technology, but what are these different applications? So these kind of different parts of the idea or different ways that the idea is expressed. And he says, you got to test it in the actual system, meaning in eukaryotic cells. And Dr. Church, who is a very well-established senior scientist, has kind of tried to be neutral, very magnanimous, and give everybody involved credit. And he says things like, look, the patent fight reminds me of reading about really unhappy rich people, meaning that CRISPR-Cas9 is sort of scientific wealth that we can use to learn all kinds of new and amazing things about science, and maybe we should focus on that. So that being said, the scientists aren't the ones that own the IP rights, and they're not the ones that are battling it out in court. So we can also look at the way that the story gets told by the different institutions that are involved. And what you can see is that they don't disagree on the evidence, but there's very kind of biased framing in presenting the facts. So you can go to UC Berkeley's website and you can go to the Broad Institute's website, and each of them has a timeline of CRISPR-Cas9 and discoveries that are related to it. And if you look at the way it's presented, you can see a big difference in kind of that framing and in the words that are used. So from UC Berkeley, Dr. Doudna and Dr. Charpentier are described as changing the world. They invented CRISPR-Cas9. Whereas Dr. Zhang, he outlined a CRISPR process that's specifically for eukaryotes. And if you look at the way the Broad Institute tells the story, Dr. Zhang worked on genome editing. He's an expert on genome editing. And he was the first in eukaryotic cells, which is true, but look at the emphasis of the way that that is presented. And if we talk about the work of Dr. Doudna and Dr. Charpentier, they had some similar findings to some previous work. So I guess what I want you to take away from this is that you can look at the relevant evidence, but then if you look at the way the story is told in popular culture or in a court of law, different groups have different interests and they can frame that story in different ways. And that might affect the way that the question of who should get credit and in what form gets resolved. And now what I want to do is just take it back to the story that's the focus of our session today. And I want you to think about the story I just told, CRISPR-Cas9 and the scientists involved. And that's a modern story, so maybe it's easier to imagine those scientists, imagine all of the excitement and anticipation and pressure that they were under when they were working on solving this problem and working on getting those papers out. the publications for the original publications for CRISPR-Cas9. And I want you to realize that the scientists who were working on the question of the structure of DNA, model shown here, were in a similar environment. So it was known that DNA was the hereditary material. And they knew that it was the passing of DNA from one generation to the next that explained the inheritance of traits. So the people that were involved kind of knew that this was going to be a big deal, and that it had the potential to revolutionize science. So that's kind of the environment that they found themselves in. And the other thing is that we can take the same things, same kinds of questions and definitions, and we can apply it to this case. So we can ask things like, is the idea of DNA structure something that is novel? Like, yes, it revolutionized the life sciences. It's, like you couldn't even imagine CRISPR-Cas9 without knowing this. And we can ask, who deserves to get credit and in what form? And so with that, I would like to introduce Dr. Ram Seshardi. He is a distinguished professor at UC Santa Barbara. He is in the Department of Chemistry and Biochemistry. He's the director of the Materials Research Laboratory. And his research focuses on crystal chemistry and crystal structure. He's also taught a graduate course in ethics, unlike me. And so I think those are two perspectives that are kind of unique to guiding us in the story of Dr. Rosalind Franklin's work and her role in the discovery of the structure of DNA. And I'm really looking forward to learning about those perspectives. So with that, I would like to turn things over to Dr. Seshardi. Welcome, thank you. And I'm gonna stop my sharing so you can share your slides. Might take a second.
So thank you very much, Dr. Graves. And that was actually a spectacular presentation, I thought. So congratulations on that beautiful presentation. And I have to confess, I'm going to, as soon as this ends, I'm going to ask you whether I can make use of your slides. So I'm really delighted to be here. And I think Ashley just found me through a web search because I have some slides on the web where I speak of Rosalind Franklin, but I'm otherwise, you may quite rightly question my antecedents for being allowed to talk on the subject. I'm not a biologist, and in fact, I'm not just in the chemistry department. My primary appointment is in materials, which is in the College of Engineering. All right, so I want to tell you what I know about the story of Rosalind Franklin. And my story, I'm sorry if I sort of deviate from the script, because my story is not going to be so much about the theft of IP as much as it's going to be about the awesomeness of Rosalind Franklin. Okay, because you will notice that she didn't live for very long, 1920 to 1958. It's tragic. She died of cancer. But in that time, just the sheer volume of important work that she accomplished is just stunning. By taken in any part of the 20th or 21st century science. Okay, so let me start by actually acknowledging that the traditional custodians of the land this university has been built on are the Chumash people who still actually live here, and we are grateful to them for their custodianship, and we recognize their significance here, both in the past, the present, and in our scholarship. This is a picture from very close to my house. It's on university land. So let me start with this, that people may not actually be aware that scientific publishing goes back a very long way. Okay, so Philosophical Transactions of the Royal Society was first published in 1665, and the first editor was this gentleman, Henry of Oldenburg, and he wrote the so-called epistle dedicatory. And even more miraculous than Philosophical Transactions being published since 1665 is that you can find it on the internet, on the JSTOR website, so you can actually read this for yourself. And if you read this epistle dedicatory, which I won't sort of go through in detail, It's basically a promise from Henry of Oldenburg that if you published in this journal, he would ensure that you had priority on the work, meaning that if you published something, no one else would later be able to claim that they did the work first. Okay, so publish and perish goes back a long way. And you'd be surprised, but even in the 1660s, people were fighting about credit. They were like, no, I discovered the Northwest Passage. No, you discovered the Northwest Passage. Particularly geographical discoveries in astronomy. These were constantly like subjects of battle. So nothing's changed. So let me give you my little timeline of DNA, because in fact, you already heard a little bit about this, about how it was already known that it was the carrier of genetic information. And it's actually interesting that when DNA was actually first isolated as something called nuclein by this gentleman, Friedrich Miescher, you actually look up how he did it. It's quite disgusting, like a lot of biology, forgive me. But he isolated this thing, and then this person here, already back in 1878, which is just remarkable how advanced organic chemistry was, particularly in Germany in those days. And one reason it was so advanced was it was of great industrial importance, because they were developing synthetic dyes, they were developing synthetic rubbers and doing all sorts of things in Germany. So this person already discovered the 5 primary nuclear bases, A, T, C, G, and U of DNA and RNA. And then in 1927, this person proposed a giant molecule that could be the carrier of genetic information. And of course, giant molecules were just coming into the fore. In fact, I was a postdoc in Germany in the University of Mainz, and it was in the University of Mainz that 2 people called Hermann Mark and Hermann Staudinger first proposed the idea of polymers or high polymers, these very, very large molecules, like any plastic we now know is a polymer. It's a very long molecule. But in the 1920s, such molecules were not widely accepted. Now we know that it's not just polymers that are large molecules. A lot of the stuff of life are also these very large molecules that are basically things that are repeated. In 1928, the first clear suggestion that DNA carries genetic information was presented. And then already in 1937, someone called Astbury in the UK produced the first X-ray diffraction patterns. And this is very important. My next slide will actually explain the timeline and will give you some background on diffraction and its importance. And then, Alfred Hershey and Martha Chase showed that they proved beyond doubt that DNA is the genetic, what they knew as being derived from all these chemicals was actually the genetic material for this thing called the T2 bacteriophage. In 1953, Linus Pauling and Robert Corey proposed a model for nucleic acids already in 1953 based on some of the earlier diffraction work. But then later in 1953, Watson and Crick published their speculative model. And I want to actually be very emphatic about this. Watson and Crick did not discover the structure of DNA. They speculated. They were model builders. They speculated based on a lot of the experimental evidence at that time, some of which had been published and some of which had, of course, not been published, but they had access to. So it was very speculative. And I think it's actually a disservice to scientific history to say Watson and Crick discovered the structure of DNA. It's better to say Watson and Crick proposed the current structural model for DNA. Okay. In fact, if you read Watson's own book, The Double Helix, you see that he actually, he seemed to have spent his time in Cambridge, basically, going to the pub and doing other things that young people do. He didn't actually do a lot of work or what we'd consider serious experimental work. So this was 1953. And in 1953, they already proposed that, I should have mentioned, sorry, this gentleman, Koltsov, proposed that the molecule could replicate in a semi-conservative fashion, and I'll explain that. Watson and Crick, when they proposed the speculative model for the structure of DNA, they pointed out that the structure automatically suggests semi-conservative replication. And Messelson and Stahl, who were still graduate students at that time, the spectacular experiment. Some people describe it as the greatest experiment ever carried out in biology, where they showed, where they proved semi-conservative replication. And it was just a spectacular experiment that involved using nitrogen isotopes and density-based separations. In 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize. But before I now go back to this history, let me tell you a little bit about X-ray diffraction. Okay, because this, I always think that at every opportunity, there should be some education too. So maybe you know some of these things, but let me actually tell you about diffraction. Okay, but of course, my story is going to be also about what else happened in 1953 beyond the simplistic timeline. Okay, in 1895, Röntgen discovered X-rays, and of course, the big excitement in those days was that it could penetrate through objects. And Röntgen famously took a lot of pictures of his wife's hand with an engagement ring on, and you could see the bones of the hand and the ring, but you couldn't see the flesh. And one shudders to think about, the possibility of skin cancers having developed in on her hands in her later years. People, of course, didn't know these things. But already soon after the discovery, it was recognized that the wavelengths of X-rays are similar to the separations between atoms and crystals. And this is very, very important because crystals are periodic arrangements of atoms, and X-rays have similar wavelengths. So you could actually probe the structure using X-rays. And this was demonstrated by this gentleman, von Lohe. It's pronounced von Lohe, in 1912. He demonstrated that crystal zinc blend. I'll show you. This is the picture right here at the bottom left. Let me actually pull up my laser pointer. This is the picture right here, von Lohe's original picture of the crystal's zinc blend. And the von Lohe experiment was a rather complex experiment, but 22-year-old William Lawrence Bragg in 1912 saw the von Lohe paper as an undergraduate. It's sort of depressing to think about. And he actually had the very clever insight to recognize that these little spots around the center don't actually look like round spots, they look like little ellipses. And he knew that if you reflected light off mirrors, it can look like these little ellipses. So he said, let's treat the crystal as a stack of mirrors that reflect these X-rays. And that gave rise to the very simple law that we call the Bragg law. And this really resulted in a giant growth of X-ray diffraction, okay? So not only were Bragg, father and son, very successful at propagating it. By the way, Bragg won the Nobel Prize in 1914 when he was 24 years old, the youngest Nobel Prize to date. He got it with his father, so he helped Daddy win a Nobel Prize. But actually, Daddy was essential because Bragg wrote down the law, but his father built the spectrometer to verify it. What they also did was they built this giant school of crystallography, and it's actually no accident that there have been so many prominent women crystallographers, because many of them actually traced their scientific paternity to Bragg. The Braggs really did not distinguish between men and women in science, and they trained the spectacular array and encouraged the spectacular array of women crystallographers. In 1954, Linus Pauling was awarded the first of his two Nobels for the alpha helical structure of many proteins, which he had discovered already in 1948. So remember, I told you that Pauling in 1953 had a wrong structure proposed for DNA. But interestingly, he had already proposed that many proteins are helices. And then in 1962, John Kendrew and Max Perutz won the Nobel Prize for the structure of myoglobin, the same year as Watson and Crick. So here's a representation of myoglobin, not done using the methods that Kendrew and Perutz would have, but using modern methods as a modern representation. Kendrew and Perutz also discovered the structure of hemoglobin and actually came up with models of how the body absorbs blood, how there is a oxyhemoglobin and a deoxyhemoglobin, etc. In 1964, Dorothy Crowfoot Hodgkin was awarded the Nobel for the structure of vitamin B12. Here's a representation of vitamin B12. Vitamin B12 has very curiously a direct bond between the metal cobalt and a carbon atom. It's this molecule called cyanocobalamin, and it's a very, very unusual molecule, the first example of an organometallic bond, and it was not known until Dorothy's discovery. So in fact, I want to say something about Dorothy, which I will the next slide. But here is how diffraction works, okay? So you may say, why is he showing us a peacock? And he's actually going to tell you. So the color of a peacock's feathers are not actually intrinsic. They are what are called structural color. The hairs on the feathers have separations that are of the order of the wavelength of visible light. So every sort of particle of visible light interacts with the distances between these feathers through the same Bragg law that we use for crystals. So D is the spacing between the hairs roughly, and theta is the angle at which the light bounces off, and lambda is the wavelength. So instead of visible light and a peacock's feathers, imagine that you're looking at a crystal and you're using X-rays. So the wavelengths are not 500 nanometers, which is what they'd be for visible light, but they're of the order of 0.1 nanometer or 1 angstrom, which is typical X-ray wavelengths, okay? And the typical separation between atoms and crystals is of the order of 2 to 3 angstroms. Between two carbon atoms, it's about 1.5 angstroms, okay? So it's the same phenomenon, which has actually been known, at least the science of it has been known since the 1820s. So when Röntgen basically came up with this wavelength of energy that had the same size as the distances between atoms, diffraction just, it didn't take very long for diffraction to follow. So this is how it works. So you just take your crystals of whatever material you have, you shine X-rays at them, and you watch the X-rays come out. And in the older times, they'd come off and you'd take pictures like this. But nowadays, of course, it's all read by a computer, and computers are extremely heavily used in the processing of the large amounts of data that you get. Okay, so let me tell you about Dorothy Hodgkins. Sorry, that shouldn't be Hodgkins. It's sort of almost automatic that we tend to say Hodgkins because we all know about Hodgkin's lymphoma, which was actually named for her husband's cousin, Alan Hodgkins, who's also a Nobel Prize winner. She was the first person actually to do diffraction from a protein. So even though Kendrew and Perutz won the Nobel Prize 2 years before she did for myoglobin, she was actually the first to diffract from a protein. She was the first to solve the structures of steroids. She was the first to solve the structure of penicillin, and nobody knew about the chemistry of penicillin, but penicillin is something that has something called a beta-lactam structure. Vitamin B12 with a carbon-cobalt bond, and insulin. And it's really interesting to read about her work on insulin. She actually did diffraction for the first time in 1935. Her last paper, where she located every water molecule in this insulin crystal, was in 1988. And many of her co-authors in her 1988 paper were not even born when she wrote her first paper in 1935. I just find it astonishing. And if you read about her, it's also like a remarkable person. So it's interesting. It's interesting and important to know about Dorothy Hodgkins and other crystallographers of the time to understand that the idea of Rosalind Franklin being a good woman crystallographer was not alien to British science at that time. So let's talk about Rosalind. Okay, and by the way, it's Rosalind, like 2 syllables, Rosalind. I learned this from hearing a radio interview of Brenda Maddox, whose book I've used extensively. So in 1941, at the age of 21, she got her bachelor's degree from Cambridge, from Newnham College. She soon thereafter started a PhD with a gentleman called Ronald Norrish, who later won a Nobel Prize for fast chemical reactions, a physical chemist. And I think this is actually important. Franklin was one of the few chemists in the whole story because everyone else was typically a physicist. And Watson was a biologist. But she did a PhD basically on the physical chemistry of coals, which were very important for gas masks. Note that this is during the Second World War, right? And it's actually interesting that Norrish apparently was going through a patch of alcoholism, which you may say, why is that surprising? Because it's not uncommon for at least my limited knowledge of Oxford and Cambridge professors. Alcoholism is like par for the course. But as a consequence, she was basically almost without a guide. So she was very independent. She was just used to being independent. She soon thereafter went to Paris, where she, if you read about her life, she really sort of hit her scientific stride, because for the first time she had people to talk to. So she learned these X-ray diffraction technique and carried out pioneering work on carbons. Interestingly, she took the subject of a PhD, but used this new technique to understand it. And even today, people cite her work on the carbons, so-called graphitizing and non-graphitizing carbons. Soon thereafter, she went to King's College as a research associate. But please note that she stayed there only for 2 years. This is really stunning, So all her DNA work was done in a 2-year period, okay? And she basically left in 53 because she wasn't getting along with the boss of the lab, this guy, John Randall, but mostly she wasn't getting along with Maurice Wilkins, who later won the Nobel Prize for DNA. So she moved to Birkbeck College, which is sort of the working men's college in London. And working men's, of course, now today it'd be working persons college, but it's a sort of historically important college in London, because until colleges like Birkbeck and University College London, college education used to be very much an elite pursuit in the UK, as it is still in most countries in the world. But this person, Desmond Bernal, himself a very famous photographer and the thesis advisor at Oxford of Dorothy Hodgkins, basically encouraged Rosalind to move to Birkbeck, where she could start she could have her own, unambiguously her own research team, because this was a problem. It wasn't clear who she was reporting to, etc. in King's College. And at Birkbeck, she initiated work on the molecular structure of viruses. Okay, ladies and gentlemen, do you know what a coronavirus is? It's a protein coat around a single-stranded RNA. Do you know who first discovered that viruses could be a protein coat around a single-stranded RNA, working on tobacco mosaic virus? Rosalind Elsie Franklin, okay? And her collaborator, Aaron Klug, basically won a Nobel Prize in 1982 for the crystallography of viruses. In 1958, Rosalind, at the age of 38, died of ovarian cancer. And there are suggestions that it could have been due to, poor X-ray safety used in those days. But Rosalind was also came from a family of Ashkenazi Jews, and Ashkenazi Jews have a high prevalence of BCRA1 or whatever that you biologists should know, and that I don't need to. Okay, so here is the paper that Rosalind Franklin and her student Raymond Gosling published in Nature. This is actually 2 papers after the Watson-Crick paper, okay? So this is the photograph that apparently had already circulated in reports and had been shown to Watson and Crick, etc. But I think it's important to note the context of this work already. And by the way, she had worked on DNA. We're talking about 1953, and she had come to King's College in 1951. She had already learned how to do the diffraction while keeping the crystals hydrated. This was, nobody else was able to do this, but she brought in her chemistry knowledge. So the way to do this, and I actually didn't know this until I read this up when I was first looking into her work. You bubble air through water, and you keep the water humidified, but you then control the humidity by adding salt to the water. If you don't want it to be too humid, you add a whole bunch of salt. If you want it to be quite humid, you don't add a lot of salt. So this is, very tricky to keep the DNA crystals hydrated while they were in the X-ray tube. She also figured out that the phosphates, there are these phosphates that are a part of which is what makes it an acid. There are these phosphate groups. They sit on the outside, and that there are these A and B forms. So people have often asked, why didn't Franklin propose a structure? And this is where, of course, the Watson book almost suggests, the book Double Helix almost suggests that Franklin was not clever enough to propose the structure, okay? And I would say, ladies and gentlemen, that the historical record is completely contrary to this, because she went on to do the crystallography of single-stranded RNA in a protein capsule or capsid, which is way more complicated. So my own theory is simply that she was doing the research and she was collecting the data before she analyzed it, right? or maybe she was already analyzing it. Watson and Crick had the luxury of not doing any research at all. They were just speculating. So it was very easy for them to say, oh, here's a good structure. And there's a longer story of how much input they had. For example, they didn't know anything about the chemistry of the base pairs. They didn't understand hydrogen bonding, but they were lucky enough to share an office mate, a crystallographer who later became a professor at USC and UPenn, but someone called Jerry Donahue, who taught them all these things. So they do thank Donahue in their paper. It's also, by the way, sometimes wrongly represented that they didn't thank Rosalind. They do thank Rosalind in their paper. Okay, so this is the Nature paper of Watson and Crick with the double-stranded DNA sketched by Odile Crick, the wife of Francis Crick, who was a very close friend of Rosalind. In fact, both the Cricks were very close friends of Rosalind until she passed away, until Rosalind passed away. So they proposed the structure, and of course, they rather coyly suggested that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material, because, as all of you know, it's a double helix, and this is the semi-conservative. It's a double helix where every strand of one helix is a perfect complement to the other helix, because wherever there's A on one, there's T on the other, and wherever there's C on one, there's G on the other. So when you unravel these two strands, you can basically make two daughter strands from the original parent strand that are exact copies of the original. So this is semi-conservative replication. And as soon as they had the structure, they recognized that, well, but of course, as we've said, people had been thinking about semi-conservative replication. So this is basically the story. This is their winning the Nobel Prize. And I'll tell you why I really like this picture. But the Nobel Prize itself tells you an interesting story. So this is Maurice Wilkins, and many people ask, why did Wilkins get the prize at all? But in fact, Watson apparently started doing DNA research because he saw talk of Wilkins. And then between this proposal, 1953, and this Nobel Prize, 1963, Wilkins did a lot of the work to actually prove this double helical model is right. Remember, Rosalind had stopped working on DNA as soon as she moved from King's College to Birkbeck. This is Maurice Wilkins. This is James Watson. This is Francis Crick. This is Max Perutz, and this is John Kenru, who won the Nobel Prize for myoglobin. So Watson, Crick, and Wilkins won it for physiology or medicine, and Perutz and Kenru won it for chemistry. Now, Perutz himself has an interesting role to play in the story, because there are suggestions that it was Perutz who gave a lot of Rosalind's unpublished work in the form of a report to Watson and Crick, because they were all in Cambridge together. So Perutz later had to refute this. He was actually accused of helping an intellectual property theft, but later on in his life, it was proved beyond doubt that Perutz didn't share anything that was not already in the public domain. So really, if there is a lesson in the story, it is that don't put down anything on paper unless it's for journal submission. Okay, don't put down and circulate anything on paper. So who amongst these people is my favorite? This gentleman here. Who knows who he is? It's John Steinbeck, the literature awardee in 1963. Okay. Okay. In fact, that's the only reason I wanted to show this picture. I wanted to show that John Steinbeck is there, flanked by Watson and Crick. Okay. So how do I like to remember Rosalind? Well, I actually think that the DNA story, for those who don't sort of delve more into her life, the DNA story almost does her a disservice. It's like, oh, she was cheated of the Nobel Prize and she's cheated of the DNA. Of course, she passed away in 58 and the Nobel was awarded in 63. But, beyond that, besides that, I actually think it's good to delve into her life to find out things like this. So when Aaron Klug won the Nobel Prize for virus crystallography in 1982, this is what he said. Okay. I can claim no credit for the choice of my first subject, tobacco mosaic virus. It was the late Rosalind Franklin who introduced me to the study of viruses. It was Rosalind Franklin who set me the example of tackling large and difficult problems. Had her life not been cut tragically short, she might well have stood in this place on an earlier occasion. Okay. So I think this is my last slide. I just have these references for where I got a lot of my material, and we can talk about them. I'm trying to leave as much time as possible for discussions. And thank you.
Okay, thank you for that great talk. So I'm going to be delivering the questions. So I have a document here with a few questions. Well, I'll wait for people to see if they chime in. Does anybody want to chime in to ask some questions to our speaker? Okay. Okay.
You can ask me the big question: was there intellectual property theft?
Yeah. So some of the.
But I don't want to. Yeah.
I was going to wait for hoping to people to chime in. So some of our other discussion points are just what are the assumptions of intellectual property, and what are some of the assumptions of theft, and how does that play a role in this story? Oh, okay. So we got some questions here. Or did you want to say anything about your thoughts on that?
Well, this is the thing, right? They didn't discover, Watson and Crick did not discover the structure of DNA. They proposed a model. So if they were doing competing experiments, for example, if they were doing competing experiments on, the crystallography of DNA, and if they'd taken results from Rosalind's lab and presented it as their own, that would have been indubitably intellectual property theft, right? But literally they were not doing this. They were so it becomes much more subtle, right? So I don't want to actually address the question of was there intellectual property theft. I do want to address the question of whether their conduct was honorable. And I think the historical record shows that their conduct was not honorable.
Yeah.
That they could have done a much better job of acknowledging the results. They could have potentially suggested some joint publication. They could have done a whole slew of things which would have been more proper. But more than anything else, Watson need not have written about Rosalind so disparagingly in his book, which is really the reason why people are so angry with Watson, right? Because if you just look at the story and say Watson and Crick were model builders, they proposed a speculative model that turned out to be right. The story is actually a lot less, shall I say, bitter than how the story turned out, because Watson wrote such a sort of nasty little book, that proved for once and evermore that he's a nasty little man.
Very much so. Okay, so we've got some more questions coming in here. So was there any documentation of Rosalind's reaction to Watson and Crick's original?
So this is where it becomes really, really tricky. I thought for a long time that Rosalind was never told by Watson and Crick that they had seen her photograph 51 when they wrote the paper. That Gosling had shown it to them, but Rosalind herself did not know that they had seen it. But in fact, the truth is, Gosling told her. I've shared this information. This is also post-war British science. People were working under like great privation. Britain in the 1950s, they would have American friends send them like food packages. It was a really poor country. And certainly the science funding was not really abundant. So they believed in sharing results and so on, which they were doing. So Rosalind, I don't think actually minded. And certainly if she had minded, she would not have been friends with Crick until her last day. Okay, she was particularly fond of Odile, Francis Crick's wife, but she would not have remained friends with Crick. So Rosalind was just this remarkable woman. She just moved on. She said, okay, these guys published a speculative model that turned out to be right, but I have other fish to fry. Tobacco mosaic virus. Right?
Yeah. Yeah. So another comment is, I was surprised the Nobel Prize Committee didn't recognize her contribution. Have there been suggestion that the Nobel Committee is biased?
I mean, there's no question that the Nobel Committee is biased, but I would actually say that if you integrate the Nobel over its history, it's 120 years of Nobels now. They've done a pretty decent job on the whole, and I don't say this only because 3 of my colleagues have Nobels and I know them well, but the Nobels on the whole have done a not too bad job. Have there been people left out? Like I mentioned Michelson and Stahl. If you look at the Nobels that are being given today and you look at the Michelson-Stahl experiment, it's inconceivable that the experiment can't get a Nobel. The reason it didn't, it was these two graduate students collaborating with each other, right? So if Michelson then went on to be a professor at Harvard, I think, and that Nobels do have a way of going to professors in big universities, right? So there's a lot of lobbying. I mean, the Nobel is not like a clean thing. There's a lot of background work that goes on to get people Nobels. But I think, I will forgive them a lot for getting Doudna and Charpentier right. I was like so hopeful that they wouldn't give it to Feng Zhang as well. And I think they did the right thing. Because I did not think that Feng Zhang, did not think that the Broad Institute's behavior was very gracious.
Yeah. This is a more personal question. Have you encountered issues with intellectual property in your own research?
Actually, yeah, we have filed some patents. Look, the truth is, suggesting a structure for DNA that also suggests how life is replicated is really, really, really important science. It's the kind of science that comes along once in 100 years. Most of us, are like, scratching away at some corner of this big picture. So I've never done anything on that scale. On smaller scales, yes, I have sometimes felt that people have taken my ideas without properly acknowledging me, and but at least my own philosophy is to move on. Now, it's easy for me to say this because none of my ideas have been, here's the structure of DNA that explains self-replication.
So
having said that, I'm a big believer in publishing as soon as the work is done. Put the record out there.
Yeah, I very much agree with that. Here's another thought that maybe you or Dr. Grays could comment on. What are your thoughts about the work of Francisco Mojica on CRISPR? Do you think him being in a university outside of the US played a role? I don't know if either of you.
Sorry, what was the name? What was that name again? Is this a person from Lithuania?
Francisco Mojica.
I don't know the name. I knew the other name that was mentioned in one of your slides, Gusalis or something. I'm sorry. I don't know enough of the CRISPR-Cas9 history.
So, I mean, there are other people who were not US scientists who got credit, like, Dr. Charpentier. So I think it's more, and you kind of see this in the conflict, right? It's like, what is the discovery or the idea that you're assigning credit for, right? Is it the discovery of CRISPR as a system, or is it the translation of that into a technology for gene editing? And then, how is that gene editing applied, prokaryotic cells versus eukaryotic cells? So I think that is really, those are the kinds of things that the credit, like assigning of the credit, is coming down to. And so in the case of the Nobel that was awarded to Dr. Doudna and Dr. Charpentier, that is really for the technology. So streamlining it into a system that you could use easily for gene editing.
Okay, so one other question for the discussion. How could we?
Sorry, may I add to that?
Yeah.
Actually, an important point to note is that in the original will of Nobel, he wanted science to be useful. So the Nobel Committee in general does like things that are, go a little beyond just a lab curiosity, even if it's important. And they love the demonstration in real world terms.
No, that makes sense.
But they still give Nobels to black holes, but that's a separate matter. Yeah.
Okay, so how could we use someone else's intellectual property without getting a lawsuit from the original creators?
I'm not sure I understand the question. So if I may, because this was not so something that I spoke about so much, but as we heard earlier, you have to do a lot to prove that your intellectual property is something that is worth protecting. You have to sort of show, firstly, you have to disseminate it to the world. Typically in the US, you do that through a patent. But that's also a very interesting process, by the way. You have to show, what the prior art is. You have to show how your art is new, but you also have to show that your patent has the art in a manner that can actually be reproduced by other people. It's a very complicated and rather arcane process. So if someone else has a patent and you're copying the patent, unless they're making money on it or you're making money on it, they probably are not going to care. A lawsuit is a lot of money. They're not going to go after you if, and then of course you can claim fair use. You can just say, oh yeah, I was just doing this out of curiosity as a hobby. I wasn't making money out of it. And then they can't go after you, So these are very, very complicated legal issues.
I'll just go ahead and add, chime in here, that you can also establish collaboration agreements with individuals early on. So if you choose to use work that was already published or something similar to what you need, and it's a necessary set of research or work that could help to forward your own question, you can also reach out to the scientist and form a collaboration agreement where you discuss what expectations are going to be for usage of their work into yours, or like how you choose to move forward with future collaborated projects. So you can do that also. That's an option. But we are nearly near the end of our meeting. This is at 4:22 right now. So I'll go ahead and pass this over to Marie, but only after I go ahead and give you guys the last opportunity to answer our post-assessment polls. So remember, I'm just going to ask you the same exact questions that we asked earlier, and just want to see if your responses changed. And then we'll post the results on our Facebook page and also on our Twitter. So
I'm impressed that everyone has hung on so far. Thank you.
Thank you. All right, there you go, guys. You will have 4 minutes to go ahead and answer these questions. Choose wisely. Okay, I think that's all the responses we're going to get. So I've already closed the polling, and I'll leave it to Marie to kind of close this out.
Do you want to share the results? Okay. Okay, so everybody can see the responses now. So first off, I just want to make a few closing remarks. First off, I want to thank our invited speaker, Dr. Seshardi from UC Santa Barbara. I want to thank Dr. Grays from Auburn University for her introduction on intellectual property. And I want to thank all of our attendees for doing a Zoom at the end of a long Zoom year and being a part of our discussion, which was great, I thought. And if everybody could please use this QR code, we have actually a post-event survey. So we're going to be organizing our STEM story events once a semester here at Auburn University. And we are looking for feedback on this event to help us organize some of our future events. And on that note, we have a website, and we would recommend that you follow our social media accounts because we're going to be organizing multiple events in the future. And these are hopefully going to be in person or some aspect of in person and virtual. And we're really excited to develop and share those events for the future. So please fill out the survey, keep following us on social media, and thanks everybody for attending. Are there any other notes or points that I missed?
Okay, so the post survey won't open until after the event. So you just guys wait until that's open, but we also will be sending out the email directly to the survey via Qualtrics. So you all will still get the email. You still will be able to get access to the survey.
Okay, great. Thanks everybody. Have a great evening.
Thanks, everybody. Thank you, Dr. Seshardi. That was great.